everybody, and welcome to another episode of the SOHO Insider podcast. I'm your host, Dr. Megan Melody with Tampa General Cancer Institute, and I'm here with Professor Nirav Shah from the Medical College of Wisconsin. Dr. Shah, do you want to introduce yourself?
Yeah, hey, thanks, Megan, for inviting me on this podcast. Excited to be here. I'm Nirav Shah at the Medical College of Wisconsin. I've been here now for many years, and I focus in B-cell malignancies and cell therapy.
Thanks so much. Yeah, we're really excited to have you. Particularly, we're really excited to hear some of the updates that happened at the Tandem meeting of ASTCT and CIBMTR in February of this year. So you presented quite a lot of research, and one of the things I found most interesting was your data on your investigational dual-targeted anti-CD19, anti-20 CAR T-cell therapy, LV-2019, which I believe was used for the treatment of CLL previously treated with either a BTK inhibitor or a BCL2 inhibitor. I was wondering if you might be able to tell us a little bit more.
Yeah, happy to. I think that one of the exciting things that's happening in the CAR T-cell field is we're seeing advancement, right? We're going from our first generation single-targeted CD19 CAR Ts to an explosion of CAR Ts that are now multi-targeting. And why multi-target? Well, because people keep relapsing. And so while single-targeted CAR Ts have really revolutionized how we think about refractory lymphoma, the reality is that still many patients are going to relapse after this. So how can we do better? And so that was really the premise of this project is we have a CAR T approved with liso-cel and CLL, but we know that the median progression-free survival with that CAR T in their target population was 12 months. And so we developed an IIT using a CAR T tandem CD20, CD19 CAR T called LV-2019. It's the same construct as a different CAR T being developed that has a label of the name Zamtocabtagene. But as we did this as an IIT, we have this sort of different name for it called LV-2019. And so we looked at patients who had relapsed refractory CLL and developed a phase 1 clinical trial to see the safety and efficacy of this dual targeted CAR T in this patient population. And so we focused on those patients who had seen at least 2 lines of prior treatment. One had to be mandated to be either a BTK or BCL2. But to be honest, the majority of our patients, 83% of the 18 patients, actually were double refractory to both BTK and BCL2. We offer these patients this dual-targeted 2019 CAR T, and we also use a rapid manufacturing program where the CAR T cells were made in 8 to 12 days and given as a fresh infusion. So the idea here was to really improve on CAR T in more than one way by improving the manufacturing, by going after more than one target. In terms of our patient population, they were quite relapse refractory, I would say. The median lines of therapy was 4, ranging from 2 to 7. About a third of them had P53 aberrations. 56% had complex cytogenetics. So I think you would agree, Melody, sort of a difficult to treat group of patients with CLL. And so what did we see in terms of outcomes? I think we were really excited. We had a best overall response rate of complete remission of 78%. And so that's really exciting when we know that with the liso-cel product in that sort of similar patient population, the best CR rate was 20%. This resulted in a median progression-free survival of 32 months and a 24-month overall survival of 80% with the median not being reached. So really exciting data, I would say. Can dual targeting improve outcomes in CLL? I think we need larger studies and more patients, but this early data looked exciting and happy to be able to share that here with you guys today.
I mean, that's amazing to know that over 80% of your patients were previously treated with both a BTK inhibitor and a BCL2 inhibitor, some of our best therapies in that space, and were progressing or refractory to that treatment and still had such a remarkable overall and complete response rate. It's really impressive. And I think, we're learning, or I think we've known for a while that truthfully, time to next treatment in these patients with relapsing and recurring disease is really important in improving quality of life of our patients. So knowing that, these progression-free survival was nearly 3 years is really impressive. I would love to hear a little bit more about Zamtocabtagene. I know you also presented some data on the DAILY 2 EU study, which is a multi-center open-label study comparing Zamtocabtagene to standard of care in patients with relapsed refractory diffuse large B-cell lymphoma who were not a candidate for auto transplant. So I was wondering if you could give our listeners a bit of an overview about your presentation and key findings of this study that you presented as well at Tandem.
Yeah, happy to, Megan. And what I'll say is, so people understand the relationship that we've had as an institution is we sort of have this industry academic collaboration where we've done some IITs with this construct. And in the setting of the IIT, we call it LV-2019. because we manufacture it slightly differently. But we worked with our pharmaceutical partners, in this case, Miltenyi Biomedicine, for the pivotal studies, because that's not something obviously academics can do on their own. And in the pivotal study setting, we call the product now has a name, and it's called Zamtocabtagene autoleucel. And it's a product that's been evaluated. There's a phase 2 third line diffuse large B-cell study that's been completely accrued in the United States. And then what recently was just presented was the outcome of a randomized control trial of zamtocabtagene versus standard of care in older patients that were deemed to be transplant ineligible. So if we take a step back, when we think about randomized CAR trials, all the randomized CAR trials have only happened in transplant eligible patients, right? So this was our ZUMA-7, our Transcend study. We have actually never looked at transplant ineligible. All those studies were single arm phase 2 studies. And so I think this is an important study because older patients are at higher risk of treatment-related complications from CAR T. And so this was a randomized control trial looking at zamtocabtagene, a dual targeted CD20, CD19 CAR, sort of the theme of this conversation today, and compared to our GEMOX, which was at the time this trial opened, an acceptable standard of care. We now know that there are better regimens than our GEMOX. We've seen that with some of the new data, such as the data we've seen with glofitamab and Moz and Pola. But at the time, this was felt to be a standard of care. And so there was crossover allowed in this clinical trial, but the primary outcome was sort of the event-free survival, comparing, again, these two different approaches of chemoimmunotherapy versus going directly to CAR T, but specifically in transplant ineligible. So the first question is, what did transplant ineligible mean, in this patient population? And so it was patients who were 65 and older and had a specific sort of impairment of organ function. Any patient who is 70 years or older, or a patient who was 18 years and older and had sort of an elevated HCT/CI. So again, sort of multiple organ toxicities. And so these patients were randomized, 168 patients, 82 randomized to zamto-cel, and then 78 patients were randomized to our GEMOX. And so if we look at the demographics to show you that these really were an older, unfit population, the median age in the zamto-cel arm was 75, and the median age in our GEMOX was 74. And half the patients were actually over 75 in the zamtocabtagene arm, and a third of them had an ECOG performance status of 2 or greater. So I think that these really are patients that we would deem as transplant ineligible when we think about our transplant eligibility. What's the bottom line? There was a statistically significant benefit in event-free survival as the primary endpoint, and the median event-free survival was 6.2 months versus 2.5 months, and this was highly statistically significant, showing that even in this older, infirm population, that a high-risk therapy like CAR T was still more beneficial than chemoimmunotherapy, despite the risk of things like CRS and ICANS. They found that this benefit was across different subgroups. And in terms of the toxicity of this CAR, which is obviously very important when you think about an older patient population, almost all of the CRS was grade 1 to 2. Only 5% had grade 3 or higher. And same with the ICANS was, again, largely 1 to 2. Any grade ICANS was just 10%, and only 1% had grade 3 or higher. And with this CAR T, they saw improvements again in EFS, but also response rates, with the best ORR of 72% with zamtocabtagene versus 45% with chemoimmunotherapy. So, I think this data adds now to the entire CAR evidence data that not only is CAR better in the transplant eligible patient population, younger, fit, chemo-refractory, but now also in an older patient population. And if you really look at the demographics, like this was an old patient population with comorbidities. Zamtocabtagene was safe and effective and really showed, I think, promising efficacy. And we hope that this data, along with the data from the third line study, will actually make this a viable product and potentially approved by regulatory agencies either later this year or next.
Thank you so much. That was such a comprehensive overview. And I completely agree with you. I think that this really does highlight two strengths of zamtocabtagene. The first is that it is a good drug or a good, cell therapy construct and is a very efficacious treatment for the management of diffuse large B-cell lymphoma. And then obviously in the LV2019 setting, for CLL. But I think, as you're saying, in a broader sense as well, it helps to answer that question. A lot of the times we're getting this feedback that, oh, sure, well, CAR T-cell therapy is safe and effective, but, we have all these criteria where perhaps they would not be transplant eligible, and so previously would not have qualified for the randomized clinical trials that we have evaluating the efficacy of standard of care CAR T cell therapy. And our studies like the pilot study or the Alicante study, are often, although I very much believe in that data and utilize it to treat my patients, I think is definitely open to the fact that it's not a randomized controlled trial. And so you do question whether or not there's some selection bias in the patients that were enrolled on that clinical trial. And so this really does show, like, if we're not taking our patients to CAR T cell therapy, what are we reaching for? We're reaching for standard of care, combination chemoimmunotherapy or novel agents. And despite the advances in that field, zamtocabtagene or CAR T cell therapy, if we're applying this data more broadly, appears to be, safer and obviously more efficacious. And so I do think that really helps to answer a question that we continue to hear both in the community and academia about when to apply CAR T cell therapy. And right now, I think that this really helps to answer the question that age is not a limiting factor. I just want to take a step back a little bit more broadly and say, oh, sorry, did you want to say?
No, I was just gonna say, I think you hit it right on the dot. This is going to help the entire CAR field, right? Like, forget about the constructs, showing, because the majority of our patients are older,
Yes.
right? Like, that is our patient population, right?
Yeah.
They're 65 plus. That's when DLBCL happens. I think it's just good data to just simply show, right? And it's going to benefit Lysocell and Yescarta and all the other products because there'll be a bystander effect, right? They'll see the initial benefit to just say, look, you take old sick people and you give them a CAR and they do better.
And we've known that, but now we have the randomized data to kind of.
We have it now in the older population. We'd had it in the younger population. So I think this was a good study that should help convince, right? We're believers, right? Me and you are believers, Megan.
Yeah.
And so, like, we need other people to believe, and this helps us sell our story.
I completely agree. Now, I just want to take a step back because ultimately what you've done today is really provide some great education for myself and for our listeners about, LV 2019, zamtocabtagene, autoleucel. But I know that, you have been sort of instrumental in educating myself as well as other junior faculty on the creation of bicistronic CARs or now tandem CARs. And I was wondering if you could maybe just give our listeners a little bit of background about what you've seen coming down the pipeline in this space and what we should really be looking for or keeping our ear to the ground for in regards to advancements in the field of these dual targeting CAR T cell therapies.
Yeah, so I think this is the next iteration in CAR. I know there's a lot of things happening in CAR, allo CARs, NK CARs, in vivo CARs, but what's ready for prime time? Yeah. I think it's actually dual targeting with an auto CAR. This zamtocabtagene is one of 4 2019 CARs under development. And if you look at the data for all of them, they look to be highly efficacious, better than our standard CD19 CAR. And at the same time, they've improved the safety. So we're sort of getting we've been able to refine our CARs and make a better model, right? Which is ultimately the goal here. There's a randomized controlled trial already ongoing of 2019 CAR with a company called Lyell versus CD19 standard of care in diffuse large B-cell lymphoma. That study is going to be replicated by another company. J&J is also developing a study like that. And so there's a lot of excitement. The early phase 1/2 study looks quite promising. And then, I don't want to go into the weeds too much, but each CAR is built a little bit differently. You sort of highlighted this, Megan. The Kite sort of products are a bicistronic CAR, and they have sort of 2 separate single chain variable fragments, whereas zamtocabtagene and some of the other dual targeted CARs, they're tandem CARs. And so on one chain, they have the ability to target more than one antigen. So, this is sort of, I love the CAR analogy, right? Because you have new models, you have different ways of making the engine, you have different on-off switches. So I think really stay tuned. I think in 5 years, we'll be giving 2019 CARs, and that's good because if they do better and are safer, right? We don't want to drive around a 2001 Camry forever. Eventually, you want to buy a newer model. And so I think that's the future, but obviously the studies need to read out and show this in a randomized fashion that these are actually better.
Yeah, and I think some of the major critiques right now of our standard of care CARs is we have some constructs that are really wonderful. They're both all of them are very wonderful in efficacy, but we have some that are, probably better in manufacturing, whereas others are better in sort of safety profile. And ultimately, it sounds like most of these companies that are developing these dual targeting CARs are taking both of those things into account. So, you were mentioning LV 2019 having rapid turnaround manufacturing, but also an improved safety profile. And so some of these things where we're debating when we reach for one versus the other appears to be kind of mitigated with these newer constructs. So
Yeah,
really
I think
promising.
you have the luxury of time. They've learned from the experiences of other companies. We've gotten better at all of this. So I think that whatever comes next, I think it's going to be dual targeting. I think it's going to be faster manufacturing. I think it's going to be safer. And it will eventually displace CD19, but that's okay, right? Because we want to keep offering patients better therapies.
Yeah, always want to be innovating for sure.
Mhm.
Well, again, I wanted to thank you so much for joining us for this episode of SOHO Insider podcast, and we really appreciate your time and all of your insights. So thank you very much.
Yeah, no, I'm just lucky to be invited, Megan. So thank you. And it was always nice to see a friend and a colleague. So I look forward to seeing you at a meeting.
Absolutely.